Hello and welcome to the D21 Cell and Nuclear Division Review video. There we go. Alright, so I really like the title of this unit. I think it's very descriptive. It's showing us right away that there's two types of division, and we're going to learn about both of them. We're going to learn about even more than that. So standard 1 says generation of new cells in a living organism by cell division. So we have this concept here. We're starting with cell division. So the only way to get new cells is through cell division. New cells. And so you take a parent cell, and that parent cell will split into two what we call daughter cells. And this is really important distinction, right? It's always 1 to 2. So you start with 1, and then that one will split into 2. It's never splitting into more than that, right? It's splitting into 2. In the guidance it says that this is sometimes also, instead of parent cell, called the mother cell. And it gives two daughter cells. Although one of these, right, like these were just one cell that got bigger and then split in half. And so how does it actually split? That's standard 2. The process of it splitting is called cytokinesis. Cytokinesis. So cytokinesis is our vocab word that means splitting of the cytoplasm. Between daughter cells. So you're going from the parent cell to the daughter cells, and cytokinesis is how you split the cytoplasm. So cytoplasm is that stuff on the inside, and you've got to split it somehow. And so depending on what you are, it splits differently. If you're an animal versus a plant, the processes are very different. They're almost inverses of each other. So in an animal, the cell is really large, and it starts to pinch inward in what they call a cleavage furrow. So it's pinching inward until it pinches basically all the way in, and then they separate like this. This is driven by these things called actin and myosin. So actin and myosin, they're contractile, so they contract. Contractile. And so what they do is they are causing the cell to move inward and pinch. And so they, they actually form the cleavage furrow. And so they're a ring around the outside. And so they would be like, if we were to draw it 3D, and they're pulling it inward. And so that's the actin and the myosin are creating this contractile ring. And so they are able to do this because they push and pull on each other, and so when they pull, they're pulling the cell membrane inward. So they're pulling the cell membrane inward. So that the black solid line is our cell membrane. So they're pulling the cell membrane inward until it splits. And so that is cytokinesis in animals. You've got the cleavage furrow, the contractile ring, and then it's pulling it inward. In plant cells, it's almost the exact opposite. You have your cell, but in plants, they have a cell wall. And so you've got your really big cell, and what happens is you actually will basically create the cell wall. Oh, sorry, I did not mean for it to get smaller. From the inside out. And so you can see that it's slowly increasing in size. And so what you have is you have vesicles. So vesicles, remember, are little membrane-bound particles. Your vesicles are going to cause this. And so the vesicles are going to come and they're going to add more, until it is all the way built. And so this vesicle secretion is going to form what's called the cell plate. And so the cell plate will eventually give rise to the new cell wall. And so you have, over here you have a cleavage furrow, over here you have the cell plate, and so you can see how this one is pinching inward, whereas this one's growing out. And that is going to lead to cytokinesis in both of them. So you end up with two cells. But in plant cells, they're stuck together because of that cell wall. Alright, standard 3. In standard 3, what we have is not all cells divide equally. So sometimes we get like one that gets most of the cytoplasm, and the other one just gets a little bit of the cytoplasm. So most cells, it's equal, but in some, it's unequal. Those are our exceptions. So our first one here, our first exception we need to know is oogenesis, which is a really big word that all it means is production of eggs in humans. So our egg cells are also called oocytes, and so when we produce egg cells, only one of them is big and the other one is small. And so we'll learn about why that is another time. And so that's in humans. In yeast, which is a type of fungi, we get budding, and so the bud would look something like this, where it looks like that, and then it buds off, and so it looks very similar. So yeast budding also does unequal division of the cytoplasm. Alright, so that's cell division. Moving on from cell division, we have nuclear division. And so nuclear division is always going to come before cell division, because you have to create the new nuclei for each cell. So that you need a new DNA that's identical, depending on what you're doing, in each cell. And so here we have, and for most of these next standards, we have mitosis and meiosis. And they're different processes. So mitosis is going to create identical nuclei, and meiosis is going to create nuclei with half the DNA, because these are going to give rise to sex cells, and this is going to give rise to just your somatic or body cells. So you have the two different ones, and so standard 4 is about the difference. And so mitosis is maintaining chromosome number and your genome. So you keep your genome intact. And this one is going to half it and create genetic diversity. And we'll learn more what that means in the later standards. Okay, so that's standard 4. Standard 5. How do we make sure we have identical ones? Well, we do DNA replication. And so DNA replication happens before mitosis and meiosis. So it has to happen before. So it's a prerequisite. And so then what you have is you take your DNA and you duplicate it and you get chromosomes. And now chromosomes is one of those weird words where it can mean different things depending on what part of the process you're in. And so it looks like this, and it's held together at the centromere. And so this is a chromosome, but it's duplicated. And so before you duplicated it, it was a chromosome, and it was just the one. That was a chromosome. But because of vocab and the way we do it, this is called a chromatid. It used to be a chromosome, then you duplicated it, now it's a chromatid, so it has a new name. And so these together, both of them, are called sister chromatids, because they're identical at this point. And two sister chromatids make up a chromosome. And so these will stay together until later in the process, and then we split them. Alright, then, to do this, to have these chromosomes, we have to condense them. And so they undergo condensation, and that's just a way of saying they package more tightly together. And so they're packaged by histones. If you're in HL, you already learned about histones. Histones are proteins that DNA wrap around. So they're proteins that DNA wraps around. And if you're in HL, you know that those form nucleosomes. Histones. Okay, and so this process here is called supercoiling. Supercoiling. Once you've condensed them, you need to move them. And they're moved the same way in both mitosis and meiosis. They're moved through these things called microtubules and microtubule motors. I don't know why, but I always find that they look kind of like pickup sticks or like K'NEX toys. And so they're these like rods that you find. So they're these tubes, and they're found all throughout the cell, and they help move it. So they like attach to the chromosomes, and they are responsible for moving them. And so they look kind of like these like swirly hollow tubes like that. And I don't know, they look like toys. It's kind of weird. And so they move them. We have our phases of mitosis. So, okay, we've got it. They're condensed. They're ready. Then we can move them. We've got our microtubules. So now we have the actual phases. So inside of mitosis, we have 4 phases. We have prophase, metaphase, anaphase, and telophase. And it's often abbreviated PMAT. PMAT. And so in prophase, they condense into that X, and then the microtubules start to form. In metaphase, they line up in the center of the cell. And so you get, for mitosis, they line up like this. You've got your microtubules attached to them. So it looks like that. There we go. And so this would be metaphase. They've lined up in the center. Then for anaphase, you split them, and you break that connection. So you take your 2 chromatids, and you split the chromatids, 2 at either side. So it looks like this. You're dragging them to either side. You ripped it away from its best friend. So these identical chromatids have been separated. And then in telophase, the nucleus starts to reform. And they start to decondense. And so it looks like this. You've got like, oops, like that. So the nucleus is starting to reform, and they've been separated. You need to know what it looks like under a microscope. And so yeah, standard aid is just, can you view them under a microscope? And it would look kind of like this, but like for prophase, it's typically like, you see like a bunch, and they're kind of like spread out. In metaphase, you're looking for that more traditional down the middle. Anaphase, they're separated but not all the way, and then telophase, you're kind of, you should see 2 clumps like that. Alright, standard 9. Meiosis is a reduction division. It's called a reduction division because it's reducing the number of chromosomes from diploid, which is 2 sets, to haploid, which is 1 set of chromosomes, where N is the chromosomes. So depending on the organism, that N is different. So for humans, N is 23. For chimpanzees, it's 24, and we know why. Fusion. Alright, so why do we do meiosis? We do meiosis because it's important for a sexual life cycle. And so that's what most animals have, is they do sexual reproduction. And so if you're going to take an egg cell from one thing and a sperm cell from another and combine them together, you can't have all of the chromosomes. If you did, you'd end up with this like exponentially growing number of chromosomes, and that would be really bad. So instead, if you want 46 total, you need like 23 in each. And so that's the purpose of meiosis, is to give you these gametes or sex cells that you can then use. You're supposed to be able to outline the 2 rounds of division. So in my mind, what that means is round 1, you split homologous chromosomes. Or I'm going to use the word separate. I think it's a better word. Separate homologous chromosomes. In round 2, you split the chromatids, just like you do in meiosis. So round 2 looks very much like mitosis. Round 1 looks different. In round 1, they line up like this in pairs of homologous chromosomes. And then one will go to one side and one will go to the other side. And I'm going to skip to standard 11. We'll come back to, well, yeah, we're going to skip to 11 because it's variation. How is it creating variation? Well, one of the ways is right here. If you have these lining up in the middle, and I'm going to redraw it in color. If you have like this, this, and this, where each one represents a different like source of a chromosome. So you are a combination of genes from your mom and your dad. So for example, let's say that this represents from your father, and this color represents from your mother. What we can see here is that this one will be 2/3 from your father, 1/3 from your mother, and this one will be 2/3 from your mother, 1/3 from your father. This is called random orientation. Random orientation of bivalents. And so these like each set is a bivalent, and it's random which side they end up on. And it's random which one they end up in. So if you imagine that that happens for each of the 23, you get this new variation. So it's partially from your father, partially from your mother, and then that means in terms of the offspring, it's partially from their grandfather, partially from their grandmother. So they get this new combination that has not been seen before. Now, the other way that it happens is crossing over. So in prophase 1, so this one happens in metaphase 1. Spelled metaphase 1. In prophase 1, when they're like paired up, you can have them cross over. And what that means is they're going to exchange part of their DNA. So this one now has a little bit of each on it, and you can see that it is going to give us 4 unique chromatids. So crossing over gives us 4 unique chromatids. Without crossing over, you would only have 2 unique chromatids. And so it allows even more variation. So random orientation and crossing over both give us lots of new combinations of alleles. It's all about changing combinations of alleles. You're not really doing mutations here, you're doing new combinations of alleles. But speaking of mutations, we do have chromosomal mutations such as non-disjunction. So this is standard 10. So non-disjunction is when the chromosomes fail to separate. And it can happen in meiosis 1 or meiosis 2. So it can happen in either part, the first round or the second round. This one, meiosis 1, is a little more likely, especially depending on the age. And so our main one that we know about is called Down syndrome. And Down syndrome is also known as trisomy 21 because you have 3 copies of the 21st chromosome. And so basically, instead of you pulling the one this way, you're going to accidentally pull both of them to this side. So then you end up like this, and so then in the second round they would line up like this, and you would split and you would get an extra copy in each of these cells. So now you would have 2 instead of just 1 copy like you should. And then you combine that with like a sperm cell, and you end up with trisomy 21. So that is an error in meiosis. Alright, that is the end of SL. So now for our HL only standards, we have cell proliferation. Cell proliferation is just a fancy word that means growing lots of cells. And so some of your cells barely ever divide, some of them never divide, but some of them grow a lot. And so they can grow a lot for different reasons. One of them is growing. So when you're in like an embryo, you'll experience a lot of cell proliferation because embryos need to grow rapidly. The other spot where we see growth happening is plant meristems. So plants basically grow and grow and grow. They have these regions called meristems. Those are found in the roots and in the shoot or the stem. And when they do that, they grow and grow and grow, and that's how trees get taller, bushes get taller, etc. Now, in addition, you also have cell replacement and tissue repair. And so you have cell proliferation and growth, but you also have it for replacement and tissue repair. One of the main places you have it is skin, right? So your skin is constantly replacing itself. And then tissue repair is from like any kind of wound you might have, so wound healing. So skin is done for cell replacement, and then tissue repair is any kind of like wound healing. And so your skin is like replacing itself really, really frequently, so you have a lot of cell proliferation there. These are spots that happens a lot. Now, this next standard, I'm not sure why it's not an SL standard 2 anymore, but it's the cell cycle, and it's basically just, can you put it all together and know all of the different parts? And so you've already learned 2 parts. You learned mitosis and cytokinesis, but we didn't really talk about interphase. We talked about DNA replication, but not interphase. So there is this other phase called interphase, which is like between, right? So interphase composed of 3 parts, G1, S, and G2. G1 is called the gap 1 phase, and here you have growth of the cell. It's getting bigger, it's doing metabolism, it's doing all kinds of things, just kind of living its life. Here is the synthesis phase, and this is where DNA replication happens. So it's the synthesis of DNA. And then in G2, you have like more growth, more metabolism, and duplication of organelles. So you're going to make more of the organelles that need to be made through duplication. So for example, the mitochondria and the chloroplast. And so then from there, you have mitosis and then cytokinesis. So the 3 like main parts are interphase, mitosis, and cytokinesis. Interphase has G1, S, G2. Mitosis has PMAT, and then cytokinesis is just, it's singular states. There's nothing else with it. And so we talked about this already. I guess that was 14 as well too, is interphase is when it's active. Now what actually controls this is called cyclins. So your whole cell cycle is controlled by cyclins. And so cyclins, if this is their concentration, they'll go up and down depending on where they are. And so these different ones, when they hit a threshold level, they'll trigger the next stage. So for example, this one might trigger mitosis. This one might trigger cytokinesis. And so cyclins are proteins that control the cell cycle. And so they need to reach a threshold. And when they reach that threshold, they trigger the next part of the process. Alright, now, there are genes that control the cell cycle, including cyclins. Now, there are special genes called proto-oncogenes, and these are the ones that control the cell cycle. When they're proto-oncogenes, these are the normal ones. A mutation in these genes caused by a mutagen will lead to an oncogene. oncogenes cause cancer, or they lead to uncontrolled cell division. And so they lead to a tumor is a better way of phrasing it. And so there are, and then that leads us to 17. There are different types of tumors. There's two main ones. There's benign and malignant. And so a benign tumor is non-cancerous. So it's a cell growth, but it's not cancerous. And a malignant tumor is cancerous. And then for the malignant ones, you have two types of those. You have primary, which is where it starts, and then a secondary tumor has moved from its original site, secondary site. So now it's somewhere else. So you have primary and secondary tumors. And so when they move, then they can get stuck somewhere else, and they can start to grow somewhere else too. So they're often moving through the blood or the lymphatic system. And so when they're moving, then they're at risk of damaging other organs. And so you can find these in like the lungs, the liver, the kidneys, etc., lymph nodes. Those are common places that you'll see those. Primary tumor sites are places like the skin, lungs, intestine, colon, areas that are subject to mutagens at high levels. But technically primary tumors can be found anywhere depending on what the mutation was that caused it. And that is D21.